And our next speaker is Karin Kiontke. She's from David Fish's lab at NYU.
So yeah, I'm going to report on a collaboration with the lab of Doug Portman, with some contributions from Eric Schwartz. And so the development of C. elegans from L1 to adult is regulated by the heterochronic pathway, which has been elucidated mostly by studying seam cell development over many years. And here you can see an abbreviated version of it. I added set 3 here. And for the purpose of my talk, however, we can focus only on this part, the L4 to adult transition, which allows me to simplify this pathway quite a bit. During the L4 to adult transition, a whole lot of interesting and important things happen. The seam cells exit the cell cycle, they fuse and they produce the ali. The hypodermis makes an adult cuticle and initiates the last molt, the final molt. The vulva undergoes morphogenesis, and we are interested in males. So in males, the male specific structures in the tail form. We can also ignore most of these and focus only on the tail tip, which is long and pointy throughout the larval stages until in L4 those cells round up under the cuticle. You can see this here. They also fuse, they migrate anteriorly to form the short and round tail tip of the adult male. This tail tip morphogenesis is also under the control of the heterochronic pathway. We know that these genes are involved, and we know that from studying mutants. For instance, if we are looking at a LET7 mutant, tail tip morphogenesis does not happen at L4, so that the adult males have this long tail tip sticking out. This is a retarded phenotype. The tail tip will retract in adult. Conversely, if we are looking at LIN-14, LIN-28 or LIN-41 mutants, tail tip morphogenesis happens one stage too early in L3, so that the L4s already have a rounded tail tip, and this continues through the L4 stage so that the adults have these over-retracted tail tips. That pathway feeds into a transcription factor, DMD3, which is the master regulator of tail tip morphogenesis. So looking at this process, we identified 2 new heterochronic genes with a very penetrant retarded phenotypes. In these 2 genes, LEP2 and LEP5, the tail tip is entirely unretracted, so you can see this here, long and pointy. However, it does retract in adult males, which shows that LEP2 and LEP5 both are bona fide heterochronic genes. Surprisingly, there is absolutely no seam cell phenotype. They are totally normal in LEP2 and LEP5 mutants, although we know that this, the effect of these genes is not restricted to the tail tip, as they also undergo an adult molt, which shows that the hypoderm is affected. I can talk about LEP2 and LEP5 together here most of the time because they have an extremely similar phenotype, except that LEP2 males also have a defect in mating behavior, which we don't see in LEP5. We wanted to put these genes into the heterochronic pathway, doing genetic and expression epistasis analyses. We found that LEP2 and LEP5 act upstream of DMD3, of LIN-41, and LET7. We further found that LIN-28 is epistatic to LEP2, so I'm showing here an example for LEP5. So knocking down LIN-28 with RNAi has the same phenotype in wild type as it does in LEP5 mutants. Knocking down LIN-14 has no effect on the tail tip phenotype, so we conclude that LEP2 and LEP5 both act upstream of LIN-28 and either downstream of LIN-14 or in parallel. So we wanted to know how LEP2 and LEP5 affect LIN-28, and for that we looked at a LIN-28 protein, at the LIN-28 protein, with a reporter construct and with Western blot. So in wild-type animals, LIN-28 is expressed in L1s and is then downregulated, which has been nicely quantified by Stadler et al. You can see in later stages there is pretty much no LIN-28 seen, so also here, but not so in LAB2 or LAB5 mutants. The reporter is still expressed in L4 animals, and here the endogenous protein is also upregulated in later stages here in L3. We next looked at messenger RNA levels. The messenger RNA for LIN-28 is reduced by a factor of 5 after the L1 stage, and that downregulation is normal in LAB2 and LAB5 mutants, as you can see here. So both of those genes regulate LIN-28 post-transcriptionally. It could do this by two ways. It could either block translation or it could be promote degradation. So to distinguish between these possibilities, we tagged LIN-28 with the photoconvertible Dendra2. This reporter fluoresces in the green range, and in L2 animals it's particularly bright in the neurons in the pharynx region, which you see here. If you expose this reporter to a short wavelength light, Dendra2 undergoes a conformation change, which is irreversible, and now fluoresces in red. And this allows us to follow the fate of this LIN-28, which was present in L2 stages, which is now red, and newly synthesized LIN-28 protein would now be green. So we did this, and we looked at the same animals the next day. Now there are L4s. In wild-type animals, you see very little fluorescence, which is expected, because by this stage, LIN-28 should be mostly gone. However, in mutants for LAB2 and LAB5, this red version of LIN-28 is still there, which means that it hasn't been downregulated as in wild-type. And we don't find any newly synthesized LIN-28. So obviously both of those genes affect protein degradation. They promote protein degradation. So now what are they? We mapped our mutants for LAB2 to the 4th chromosome, to a gene which is homologous to human microRNAs. Those proteins have a typical arrangement of motifs, several zinc fingers here in blue flank a ring domain. And that points to the function in humans where there are 3 paralogs of microRNAs. It has been shown to act as an RNA binding protein, zinc fingers, right, and an E3 ubiquitin ligase. We have 5 different alleles. One is a complete deletions, other are partial deletions. We also have point mutations only in the zinc finger. They all have the same phenotype. Those microRNAs are conserved across eukaryotes, and we think that the biological role of microRNAs may also be conserved. In mammalian or in humans, microRNA3 variants are associated with defects in pubertal timing, and puberty is the human juvenile-to-adult transition, just like the L4 to adult transition in nematodes. So we think that microRNAs could be part of this conserved axis, which we already saw in Benjamin's talk, between LIN-28 and LAD7. It also includes LIN-41, and it is important for transitions from undifferentiated to differentiated states. So now what is LAB5? We have 2 mutants, and those were mapped to a gene H36 L18.2 on the X chromosome. We have a large deletion, which takes out the entire gene, and 1 point mutation. And there's not much known about this gene. It is SL1 spliced, polyadenylated transcript is a little more than 900 nucleotides. There is a short ORF predicted in worm base, but the function is unknown because it's not conserved. However, it is the sequence is well conserved across Caenorhabditis species. So you can see this here in a screenshot from the UCSD genome browser. Blue and green indicates conservation at the nucleotide level. Surprisingly, there's no conservation at the protein level. So there's no conserved protein or peptide. So we were thinking that this may actually not act as a protein. To show that, we fused the C. elegans promoter to this Brixey sequence, which has no ORF, and expressed that in our mutants, and here you can see the result. This construct rescues as well as the wild-type construct, or as the C. elegans version of this gene. We also introduced 2 and 3 stop codons into the predicted coding region, and that also rescues. So it doesn't seem to produce a protein. Looking at modern code data, we see that LEP5, the full-length transcript, is highly expressed after the L1 stage, but in the small RNA reads, there is no signal, so we think that LEP5 is not processed into a microRNA, but functions as a long non-coding RNA. So to begin addressing the function of LEP5, we used TurboFold and the information from 15 Caenorhabditis species to predict the secondary structure. The black dots here show conserved positions in at least 10 species, and we do see some conserved structures here, of which this one is particularly interesting because this is where the point mutation is located here. TurboFold predicts a pretty dramatic change in the structure when this mutant is mutated. And in silico, we can restore the structure by introducing a compensatory change right here. So we wanted to know whether that is also true in vivo. Did rescue experiments. We already know that the wild-type LEP5, which has this structure, rescues at 0.5 nanograms per microliter. If we are injecting the FS8 or mutant version, it doesn't rescue as well, although there's a little bit of rescue. You can see this here. That is not unexpected because this is a temperature-sensitive mutant and there's not a null. If we introduce the compensatory mutation, which restores the structure, the rescuing ability of this construct is also restored. So clearly the structure of this area here is more important than the sequence. So we were thinking about this and also that this gene functions in a very conserved pathway, wondering whether we really can't find this outside of Caenorhabditis. So it is easy to find LEP5 within the species which are most closely related to C. elegans, where it's 70 to 82% conserved, but using BLAST, we didn't find it anywhere else. So I did a more careful search in the synthetic region of Caenorhabditis angaria, which is here, and I found a sequence which is has the conserved introns. It is it produces a transcript which is SL1 spliced and polyadenylated. It's 54% similar to C. elegans. We reconstructed the structure. It's not very similar, but as you can see here, it has that stem loop. So we expressed this in our mutants, and to our delight, we saw that it actually rescues the phenotype. So this is Caenorhabditis angaria LEP5. We think that we can't find this anywhere else because it is difficult to use sequence similarity alone to identify this in other genomes. So, and the very last slides I have, in 2 minutes, not even, I'd like to spend on to try devise a model for the molecular function of these genes. For LEP2, we think our best hypothesis is that this acts as an E3 ubiquitin ligase, like a human microRNA3, on LIN-28. For LEP5, it could, like many other long non-coding RNAs, act as a transcription regulation. In that case, we would expect it to be expressed in the nucleus, and it would also have to act on LIN-28 indirectly. However, LEP5 is extremely highly expressed in the cytoplasm, and it's more or less excluded from the nucleus. It's also expressed in the same tissues where LIN-28 protein is found. It is expressed, however, it's upregulated. The other expression is upregulated when LEP5, when LIN-28 goes down. It has been found to directly interact with LIN-28 in a ClipSeq experiments. So we think that LIN-28 and LAB5 interact directly. And here's my model real quick. We think that LAB5 is is acting as a scaffold to bring together LIN-28 and LAB2 and allow LAB2 to ubiquitinate LIN-28. And in earlier stages, this is not happening because LAB5 is not there, although LAB2 is present the entire time. So here, SET3 is doing the job, and we think that LIN-28, at least in the tissues where it's expressed, where we see an effect of LAB5 and LAB2, is doing the job in later stages. So with this, I'd like to thank people who contributed reagents, students who helped with this, and the CGC. And I guess I have 30 seconds for for question. Thank you very much.
I'm afraid we're out of time, so we'll go to the next speaker. Please save your questions for later. Our next speaker is going to be Julie Claycomb from her own lab at UToronto.
Okay.